So when we talk about hydrogels, we can talk about materials with a number of different structures. And the two rows of pictures here are meant to represent chemically cross-linked hydrogels on the top and physically cross-linked hydrogels on the bottom. So the chemical cross-linking, of course, is covalent bonding, and you can have sort of perfect networks on the left, you can have imperfect single networks in the middle, or you can have the ever popular at the moment double network gels, where you have two independent networks that are not actually linked to each other. All the cross-linking points are between the single components to their own same part of the gel. On the bottom, we have physically entangled gels on the left. We have special gels in the other two cases, and these are actually special gels that we've played with. In the middle, you have something that looks like the structure of a gelatin gel, where you might have little domains of interaction that form little helices. And on the right, you have alginate, which is when you have the cross-linking due to a divalent ion, like a calcium ion, where the calcium ion actually grabs onto two chains and you get little local interactions. And it can be multiple interactions like shown there with 2 or 3 individual points. So as is quite obvious for a conference in indentation, we're going to be talking about indentation. And in this talk, I'm going to talk exclusively about spherical indentation, because of course we have materials here that you wouldn't expect to have anything like plastic deformation. And so we're going to be basing all of our analyses on the elastic solution of Hertz. And we're going to do time-dependent experiments, and they're largely going to be in displacement control. So on a nanoindenter, that involves using feedback, since nanoindenters are by default load-controlled instruments usually. And so we're going to use displacement control, ramp up, hold, and when we do that, we're going to see load relaxation. And we can fit the load relaxation data to one of 2 different phenomenological models like viscoelasticity or structural models like poroelasticity. And I'm going to be talking a little bit about both, because of course we can fit the same data to either model. Now, that's an interesting point, because the behavior of the material itself could be viscoelastic, it could be poroelastic, it could be poroviscoelastic, and of course we can fit the data to the numerical models regardless of whether it's the appropriate choice for the material of interest. And so that's a key interesting point to think about going forward, that we need to actually think carefully about which is the most appropriate model to choose for fitting the data. Now I'm going to argue that viscoelasticity is a phenomenological model, but it's actually a very useful first go for establishing the extent of time dependence, or how important the time-dependent deformation actually is in the grand scheme of things. So if you have an elastic solid, you can think of it that its elastic modulus at very long times is the same as its elastic modulus at very short times or instantaneously, and so therefore the ratio of those two things is 1. If you have a fluid, whether it's just a Newtonian fluid or whether it's some sort of a viscoelastic fluid, if you take that same ratio, it'll end up being 0, so no force can be sustained in the long term. So a viscoelastic solid, by definition, is something for which that ratio is in between 0 and 1, and if it's closer to 1, it's closer to an elastic solid. If it's closer to 0, it's closer to a fluid. So that ratio is actually really interesting, because we can use that to characterize how much time-dependent behavior we have, or how important the time-dependent behavior is in the overall grand scheme of things. And you can then do things like, you can take 2 materials. Here I've got ones that have the same instantaneous modulus, but they have different long-term equilibrium moduli, so then that ratio would be different, even though one of the endpoints is the same. And you can do the opposite. You can pick materials that have the same equilibrium modulus, but that have different instantaneous moduli, and therefore different overall ratios of that viscoelastic fraction. So as I mentioned in my talk yesterday, we set up a system whereas we do a lot of our measurements twice, once on a nanoindenter and once using what we call a microindenter, which is really just a low-force instron machine set up with a 5 Newton load cell and a bunch of reasonably large-scale spherical probes. Now the reason we started doing this in the first place was when we started doing nanoindentation of these hydrogel materials, for which clearly these machines were not designed for, we wanted to get an idea as to whether our data were actually quantitative. So we wanted to see if the modulus values we got when we made macroscopic measurements were anywhere near those that we got when we made these very small scale measurements, where they could be prone to so many different errors due to the fact that the machine design is not optimized. So when we do that, and I'm going to start here with a very reasonably boring and I think actually good candidate for standardization gel, which is a 20% of polyacrylamide. So we chose 20% because that gives it a similar composition to a lot of soft biological tissues with 80% water. And it's a very formulaic standard thing to make up with recipes. It's pretty repeatable, it's very robust. We've made many of these things over the years and we usually get the same answers when we test them in many different ways. So what I'm showing here on the left are the load relaxation curves for these two different setups where we have nano indentation at very small scales and microindentation at much larger scales. And these are plotted over the same time frame, so ramping up to the peak in around 5 to 10 seconds and then relaxing for the same amount of time. Now, there's an interesting point here that you see that the relaxation behavior look very different. And several people alluded to this yesterday, that there are length scale effects inherent in these materials. And I'll come back to that. But for the moment, I just want to focus on one aspect of these gels, which are that these chemical gels that are covalently cross-linked have a very high elastic fraction. So they are something on the order of 90 to 92% elastic. They have relatively small dissipation, and in the case of those data I just showed in the time frame for the microindentation, even less so than in the case of the nanoindentation. So you can see here what's plotted on the lower right are the instantaneous and the equilibrium modulus values, and from those you can see that for these hydrated gels you get about 0.9 to something like 0.93. The other two sets of data on there, just in case you're interested, where we were playing with what happened when we dehydrated something just by leaving it out to dry in air so that there was just no water in the hydrogel, versus if we actually exchanged the water out for ethanol and therefore had a dehydrated gel that was still sitting in a fluid environment. And as you can see, we get an increase in stiffness of about 3 orders of magnitude in either case. That's kind of a topic for another story, but it's a really interesting and kind of fun experiment to do. Now, those were chemical gels with their very elastic behavior. These are a couple of physical gels. We've got agar, which is seaweed derived, and gelatin, which of course is hydrolyzed collagen. Now what we've plotted here is 3 different gels. So for agar here we have 1%, 2%, and 3% solids. And for gelatin, 7.5%, 15%, and 30%. And we chose these 2 sets after testing many different percentages of gels, because actually the equilibrium modulus values, if you look at the lower levels on the agar plot, are similar to the lower levels on the gelatin plot. So what we have are equilibrium modulus essentially matched, but instantaneous modulus quite different, because the agar relaxes quite a lot, so is much more fluid-like, down to about 0.3, and the gelatin somewhere between 0.8 and 0.85. So we have materials for which the elastic properties are in some way similar, but the time-dependent behavior is quite different. So what are we going to do with these? Well, we're going to play. We're material scientists, so we want to see if we can make materials with a range of behaviors and then characterize those. And of course, because we are doing this in a bioengineering context, we're thinking about how do the cells respond differently to changes in the elastic properties versus changes in the time-dependent behavior. So we took and made 2 different set of mixed gels. So we took our stiffest gelatin and our most compliant agar and made different combinations of those, and similarly did the opposite and took our most compliant gelatin and our stiffest agar, and we made these in 10% increments. So 10% of the 30% gel mixed with 90% of the 1% gel at the melt phase, stirred them really thoroughly, and these are thermosetting gels, so then let them sit. And what you can see across the bottom there are from 100% agar on the right to 30% gelatin on the left. So those are the 100% extremes, and then 20, 40, 60, 80 across there of agar. So that's how we're going to think of these. They're composites. We're mechanics people as well as materials people. So we're going to plot things like a composites person would as the percentage of one gel versus the other gel. So there's a lot of numbers to keep straight here, but we're going to forget now about the details of what the percentages are of those 100% extremes and focus on the proportion of those. So here's some interesting data on gel swelling for these 2 different systems. So for each one, we made these gels every 10%, and then they're plotted there as the volume fraction of the gelatin melt component. And what you can see is that we get two very different types of behavior for these two sets of gels. So we have on the top, when we have a very stiff agar and a very compliant gelatin, we have very little swelling until the gelatin fraction gets to be significant. Whereas when we have the opposite case, and we have a lot of gelatin and a more compliant agar, we get essentially a linear increase in the swelling. So this is starting to give us a hint that there are some interesting things going on. And remember that we had very different time-dependent behavior for these two sets of gels. So if we plot the equilibrium modulus on the top, and remember that these two gels were picked so as to be kind of in the same range of equilibrium modulus, and then we plot this viscoelastic ratio on the bottom, what you can see is that in going from about the same place to the same place about linearly across the range of these compositions, in both cases where the gelatin is the stiff component and the agar is the stiff component, we see that is looking very, very nice and mirrors the behavior, but actually we see a huge difference in the viscoelastic behavior. So those ratios being small on the left are because the agar is dominating, and then we see the smooth transition up to the gelatin-like behavior. Whereas on the right, when the stiff, really dominant gelatin is there, we get almost no contribution to the agar except in the very most extreme cases. So what this means is that we can make gels using this scheme where we can tune independently their elastic properties and their time-dependent properties. And so on the left here, we've plotted equilibrium modulus versus this viscoelastic ratio. And so we have three regimes of behavior. We basically have agar-like, intermediate, gelatin-like in the viscoelastic behavior across this same range of equilibrium modulus. And on the right, what we actually found is that if we calculated the total solid fraction of the gelatin, then all the data fell onto the same curve. So this gives us a fun sort of design space that we can use in for making interesting gels, all just based on this relatively simple curve-fitting viscoelastic model, assuming that this viscoelastic ratio tells us enough about the time dependence to give us information to compare with the elastic properties. Now if we take and go to the poroelastic case, we can do something where we get more real material information about the gels in terms of trying to get this permeability out, and from that permeability understanding about the pore size. Now, I hope it's not a disappointment that I'm not going to actually dwell on the details of how we do this, because we and others have been doing this now for quite a while, and there are going to be other talks today about this subject, and so I suspect between yesterday and today the detailed ground will be covered more than sufficiently. But I did want to add this in because of the fact that this ties in with the overall nano hub, that there are different ways of doing this, and that we and others have kind of settled on three different ways that we tend to do this. We might have some sort of a fitting function that comes out of finite element models that we can make some simplifying assumptions, and it's really fast fit and very similar to the viscoelastic case. We can do inverse finite element, which has a computational cost, but of course is very precise. Or one thing that we have done, as well as Lisa Pruitt's group out in California, is to make databases. So if we run 3,000 to 5,000 forward simulations across an extremely broad parameter space, even if the curves aren't completely normalizable as they are in the case of a fitting function where everything kind of goes onto a single master curve, we can have families of master curves in a database that allows us to cover a huge parameter space, interpolate between the different curves, and get reasonably good fidelity. When we double check by running the forward simulations back, we do get reasonably good fidelity. So those are three different ways of doing the same sort of basic thing. Now, if I go back to those gels that I was just talking about, so these are the pure agar case, the pure gelatin case, and now I'm plotting the shear modulus against the gel concentration. From the plots I showed before, you could see that they looked kind of quadratic-y up. That's exactly what they should do, somewhere between a slope of 2 and 2.5, depending on whether you're believing the solid mechanics people or the polymer physics people. And what you can see there for the gelatin is that we've actually plotted the data twice. So So the data are plotted for the solid points as the amount of gel that we put in at the beginning. But the gelatin gels, remember, do swell, and they swell significantly more the more gelatin you added. So the hollow points there are actually for the swollen gels. The agar gels don't swell very much, and so we don't have a second set of data on there. Now the reason I'm plotting them this way, and now these are the modulus values that come out of the poroelastic analysis, is that we can then take a look at how these fit in with measurements that other people have made. So we've taken and reviewed the literature for tensile tests, confined compression, indentation tests, AFM-based rheometry, everything we could come up with. And what you can see is that overall, the kind of quadratic trend is pretty good. Our data, which are circled there, are right in the middle. But if you take a single concentration, like say you take a 3% gel, you can see that the modulus values can go from maybe not quite a megapascal down all the way to maybe almost down to a kilopascal. Now there's good physical reasons for this in agar. There are not as well-defined agar components. You can have the two sugar blocks in the agar, the G's and the M's, you can buy those in different ratios. So there's no guarantee that these are the same gels. But this graph I made as part of my call for standardization in thinking that we do need to come up with a way to do better than this, both to have standardized materials that are going to be repeatable from lab to lab, and then to see that everybody's making good measurements that also agree very well from lab to lab. The interesting thing, however, and this follows on from something someone said during a talk yesterday, where the elastic properties weren't very sensitive to what was going on in terms of changes in the tissue, but the time-dependent properties were much more sensitive. If I plot that same sort of data, here's the agar and the gelatin data that we measured for our gels that I showed before. If we take and plot those same data for all the same agar gels that we can find in the literature where the permeability has been measured, actually it's much better. So that's interesting. So these include direct permeability measurements, which I'll talk more about in a second, confined compression, and indentation with a range of indentor geometries. And the one outlier there, the starred point, that value is the only case in all of these where the data were treated as poroviscoelastic. So all of these other cases, the data were treated as purely poroelastic. The interesting thing about this is that because the agar relaxes so much, it can't physically be purely poroelastic. In order to achieve that total amount of deformation and not have a negative Poisson's ratio, you would have to have some viscoelasticity in the solid skeleton. And you can actually calculate how much that should be from the basic principles. And if that's something you're interested in, I'd be happy to talk about that with you at the break. Okay, so I said I was going to come back to this question of length scales. So here are my microindentation and nanoindentation data back on our really well-behaved polyacrylamide gels. And what we can see is, as I said, we have a lot more relaxation. But of course, these are the same gels. These are the same physical pieces of gel that for these experiments we literally took from the nanoindentor room to the room where the other tester was. So they have the same material properties. They must. And of course they do. So if you take those gels and you extract the properties from either of those graphs and you put them into a finite element model, you get curves on the left that look the same. And the reason that they look the same as the experimental data becomes more clear when you look at the data on the right, where the simulations have been allowed to run for a very long period of time. So in those microindentation short time frames, we haven't yet gotten to the point where we have enough driving force for the fluid flow. We have to build up the pore pressure in order for the fluid flow to start, and that happens in a much longer time scale when we have a larger contact size. So that's really interesting, and that now brings us to my sort of closing point, which is that now we can use this for teasing apart these different mechanisms. I said that when we have agar gels, they relax so much that it can't be purely poroelastic. Now the fact that I plotted all those permeability values shows that you can fit them to a data, you can fit them to a curve, you can extract a poroelastic parameter for permeability, but that doesn't mean that you've had the right constitutive model because they exhibit too much time-dependent deformation. And we can take advantage of the fact that we have this length scale effect in poroelasticity that we don't have in empirical viscoelasticity to try and tease these two things out. So on the right here is the time constant for poroelastic relaxation. And as we saw yesterday, depending on how you write it, it's related to some square of a length scale and some modulus and some permeability. So it has that length scale intrinsically in there. And that brings us to the really interesting side point, and the reason that I keep beating myself up trying to make these measurements with a nanoindenter, even though nanoindenters are very bad at this, which is for a poroelastic material, the overall time to reach equilibrium is shorter if the contact is smaller. So if we want to be able to reach an equilibrium state and measure the equilibrium modulus, which of course is a much more stable parameter than the instantaneous modulus, then that makes a good case for doing these measurements at very small scale, which is why I'm always beating up on the instrument manufacturers to try and make instruments that are suited for this purpose, which they're not. And if I go back to the graph here, you can see that. Those two curves are just shifted, and so the nanoindentation case, you get to equilibrium at a much sooner time point than you do when you have the much larger scale of relaxation because of the larger fluid flow path length. Okay, so we said the time constant for poroelasticity intrinsically has this length scale in it. And in the converse case, the time constants for viscoelasticity depend only on the material properties. And if you go back and look at some of our old papers when we first started doing these viscoelasticity by indentation measurements, that was something that we did a lot of. If you kept the ramping time constant and then you went to different load levels, if you had a linearly viscoelastic material, you should be able to normalize those data. And sure enough, for commercial polymers, that usually works pretty darn well. And so that gives you a completely objective test for the absence of size effects or length scale effects in your material. So it gives you a lack of ambiguity. If you have a length scale effect, you have some extent of poroelastic behavior. If you have no length scale effect, you have viscoelastic behavior. If you have a length scale effect, you have to be able to then further distinguish between poroelastic and poroviscoelastic. So you need to be able to tease those two things apart. But because of the fact that you have this difference, you can do that. So because you have one set of time constants that depend only on the material properties and are normalizable, and you have another set that depend on length scale, you can use that with clever experiments, which were actually mentioned yesterday, in order to separate these things. So that clever experiment is to make multiple measurements on the gels with different size indenters. So you vary the size of the contact, but keep the indentation strain the same. The indentation strain expression is written there. So as you make the indenter bigger, you have to make the indentation depth bigger, which I've tried to draw there with my crude PowerPoint schematic. And then you can fit the data as a group. So you can no longer get the full set of material properties just by fitting a single curve, as you could for a purely viscoelastic case or a purely poroelastic case. But if you make these multiple measurements, 3 or 4 different indenter sizes, and here's where I reveal that over the course of our nano indenter plus our micro indenter, I think we have 30 different indenter sizes right now, about 8 for the nano indenter and 20 some for the micro indenter. As we fit the data as a group, then that allows us to distinguish between the viscoelastic behavior and the poroelastic behavior. So here's an example of some of our published data from this, where we've done these experiments, varying the size of the indenter by an order of magnitude, keeping the indenter depth such that our strains are constant. You can see that if they were purely poroelastic, they should normalize on the right more perfectly than they do. That's the influence of the viscoelastic. And in fact, when we started really pulling this apart, it turns out that actually the poroelastic part of the deformation and the viscoelastic part of the deformation are multiplicative, which is really interesting physics. I don't have time to go into why we think that is, but I've just plotted there the full poroviscoelastic solution, a simulation for a poroelastic and a viscoelastic independently, and then their product, which agrees with the finite element as per the expression on the right. So what you can see from these data is that in this particular set of simulations, which were meant to match up with our experiments, the viscoelastic relaxation was the faster relaxation, and the longer term relaxation was the poroelastic. And you can play with that. You can actually manipulate that. And Xuanhe Zhao has done some really beautiful work on this. If you manipulate your length scale space that you're in, you can make it such that you see one before the other. And that's actually really important because a lot of people assume that the viscoelastic is always faster, and that's not the case. It depends on the length scale, where you are relative to the length scale of the material. And the very last point I want to make is that we don't have to take these permeability values that we get from indentation tests as biblical, okay? This is a really easy thing to measure independently. So this is the rig that one of my undergraduates set up for making agar permeability measurements to validate those poroviscoelastic fits that I was just talking about. And so we've got a syringe pump, pretty crude pressure transducers, a little syringe that we've modified to be able to hold a piece of material. And all you have to do is flow the syringe pump through at a fixed rate, a couple of different rates, measure the corresponding downstream pressure, and from that then you can calculate the permeability. So you've got independent ways of verifying these things from the indentation tests. You've got compression tests to measure the modulus. You can measure the permeability directly, and these sorts of permeability measurements don't seem to be very sensitive to whether or not there's viscoelasticity in the skeleton because you do them over a long time. So we do each different velocity we run overnight so that we're really seeing the equilibrium modulus in the material, and therefore we can measure the poroelastic permeability completely independently of the indentation measurements. And we've had really good agreement for the agar, for the acrylamide gels, and for all the gels that we've tried this with. At the moment, we're working on some PEG dimethacrylate gels as well. So that is my fast and just about right on time tour through what we've been doing with hydrogels, measuring mechanical behavior via these relatively easy techniques. We do use the viscoelastic fits to give us quick and dirty information. So if we're playing with materials design, composite gels, that's really easy and doesn't require a lot of effort on our part. The size effects we've talked about, and then of course the fact that it's actually not that difficult to decouple the poroelastic and viscoelastic deformation. So with that, I'd be happy to take any questions.